Very good evening, everyone. Today we will be seeing tutorial 4 of PT39 NPTEL course. This is bioreactors. We'll get into the tutorial now. So the first question that has been given here is the degree of reduction for glucose. The molecular formula is C6H12O6 is what's the answer in the following options that have been given. So we'll just see the degree of reduction for the elemental compounds that is present in the glucose, which is C, H and O. So for C it is +4, for H it is +1 and for O it is -2. So if you just substitute it in the equation where your 4 is the, just a moment. Yeah, 4 is the degree of reduction for carbon into 6 molecules of carbon is there in the glucose, and then 1 is the degree of reduction for hydrogen, and totally 12 molecules are there in glucose. And for O2 it is -4, but here we are considering it as O, therefore -2 into 6 divided by whatever is there in the substrate of carbon has to be taken in the denominator, that is W. So by multiplying it, 24 plus 12 minus 12 divided by 6, where plus 12 and minus 12 will get cancelled. Therefore, 24 divided by 6 will be 4. So the answer for the degree of reduction for glucose is 4. Moving on to question 2. Consider the following reactions producing single cell protein by the equation. Substrate is C16H34 plus 12.5 moles of oxygen plus 2.13 moles of ammonia, giving rise to 10.6 moles of the biomass plus 5.37 moles of carbon dioxide plus 11.4 moles of water. Determine the biomass yield coefficient. Choose the answer closest to your calculated answer. Determine the biomass yield coefficient. So what we have to find here is Yx by S. So this is what we have to find. And we know the formula for Yx bar S, which is mass of biomass produced to the mass of substrate, that is euclides. Whereas in the data that has been given to us, it's just the equation that has been given. There are no mass of biomass produced and no such data has been given. So how we are going to derive the data from the equation that has been given to us? So the equation says this in terms of molar moles, right? 1 mole of your substrate is giving rise to 10.6 moles of the biomass. And we know the equation moles is equal to mass divided by molecular weight, that is weight divided by molecular weight. That is what moles are. So if we just multiply your moles into your molecular weight, you will get your weight or the so-called mass, right? So that's what we are going to do it here. So first we will find the molecular weight of the substrate as well as the biomass that has been given in the equation. So just take the substrate, which is C16 and H34. The molecular mass of carbon is 12 into 16 molecules plus molecular mass of hydrogen is 1 into 34. By multiplying and summation of it, we get 226 gram per mole. Similarly, take the biomass equation. So C is only 1, therefore 12 plus 1 into, for the molecular weight mass of hydrogen is 1 into 1.66 plus for oxygen it is actually 32, for O2 it is 32. We are going to take here O only, which is 16 into 0.27 plus for nitrogen it is 14, the molecular mass is 14 into the subscript is 0.2. By multiplying and summation of it, we get 20.78 gram per mole. So now we are going to substitute it in the final equation, which is Yx bar S is equal to, so moles into the molecular weight that we have to do here. For biomass, the mole is almost how much? 10.6 moles of biomass has been produced from the equation. So 10.6 into what is the molecular weight of biomass? 20.78 divided by how much moles of substrate has been consumed? Only 1 mole of substrate has been consumed. Therefore, 1 in the denominator into what is the molecular weight, which is 226 as per our calculation. By multiplying and then dividing the numericals, we get 0.974 gram per gram, and the answer that is closest is option C, 0.98 gram of biomass per gram of substrate that has been utilized. Moving on to question 3, which of the following is AND bar are operational parameters useful in scale-up? So I'll just tell you one thing, like during scale-up, what was given in the lecture was there are two ways of scale-up. Like you can consider two parameters during your scaling up. One can be your operational parameter that has been asked in the question. The other can be your geometric parameters. So we'll just try to rule out the options which are not related to operational parameter at all, like which cannot be changed during the operation of a bioreactor. So if you see the impeller tip speed, yeah, it's an operational parameter that can be changed during the bioreactor operation. And option B is constant volumetric mass transfer coefficient, which is nothing but the KLA, which we know the main operational parameter that is being used for scaling up. Constant is also right. Ratio of height of the liquid to the diameter of the impeller. So if you take the bioreactor, this comes under geometric parameter because the diameter of the impeller is not going to change at all. So this comes during the fabrication of the bioreactor only. So this comes as a geometric parameter. Similarly, the ratio of baffle thickness to the diameter of the tank. So here baffle is in the corner of the reactors. So the width of baffle as well as the diameter of the tank, these are the geometric parameters. Therefore, option C and D are geometric parameters, and the other two that has been given in equation, like option 1 and 2, are the main operational parameters that are useful during scale-up. Moving on to question 4, cellular energy status is defined as option A, ADP divided by ATP square, B, ATP minus ADP, C, ATP divided by ATP plus ADP. Option D is ADP minus ATP divided by ATP. So we'll just see the equation for this, where ADP plus monophosphate gives rise to ATP, right? So the cellular energy status of a cell can be denoted in two ways. It can be just product divided by reactant. So here the product is ATP divided by ADP. So this can be one of the this can be one of your cellular energy stated definition, whereas the other is kind of efficiency formula that we use. So what we use for efficiency, for example, A giving rise to B, okay, but we have to focus on product here. That's what we want to know, right? Cellular energy status. So here ATP is the main energy that is being used by the cells. So the equation for any your efficiency will be D divided by A plus B. So that's what is happening here also, like ATP divided by the total energy, ATP plus ADP, the substrate as well as the product. So when you calculate this, it will be very easy to find. Say for example, the right option here is, not for example, like the right option here is ATP divided by ATP plus ADP. It's the efficiency formula that we try to calculate. For example, if you get it 0.9 for a cell, which means 90% of the molecules are present in terms of ATP and the remaining 10 are in terms of ADP. If it's the reverse, if it's 0.1, only 10% are in terms of ATP, the remaining 90% is in terms of your ADP molecule. So the right answer is C. Moving on to question 5. Why are scale down models developed? Options that has been given: to validate the new operating procedures of a large scale production plant. Experiments are expensive to do at large scale. Option C, to produce a product of interest at large scale. D, more reliable than high scale. So we'll just see at first what is scale up. So usually what we do, like we'll just do a pre-culture from a single colony or a glycerol stock. From there we will try to inoculate our cells into 1 liter 250 ml flask or 1 liter shake flask. From there we will take it up for a lab scale reactor, which can be 5 liter reactor. From 5 liter reactor, I'll just write it down. So first from pre-culture, I'll be either putting it into 250 ml flask or else 1 liter reactor, 1 liter shake flask, which will be considered as inoculum for 5 liter reactor. From there, your pilot scale comes, which is 50 liter, and then it will go for the large scale, which can be 100 meter cube. So this is scale up. So this is what we do. Like we will see how it works in the lab scale, which is your 5 liter reactor. From there, we will develop it into devise a process for 50 liter, and then from there it will be taken it for 100 meter cube. Whereas scale down is we will be doing something in 100 meter cube and we want to change some parameters to see like what happens. So that can't be done in the large scale, right? You, for example, you have 10 parameters to check on and you can't do like one factor at a time or something in your 100 meter cube reaction, 100 meter cube reactor, where lot of time waste as well as like energy, manpower, and everything will be wasted. So experiments can't be done in the large scale. So what usually is being done is that a small scale reactor, which exactly defines the operating operational parameters of the large scale reactor will be mimicked, and then whatever the parameter has to be changed, it will be changed in the small scale reactor, seen, and then it will be again executed in the large scale reactor. So if you see the options, will come from the down, more reliable than higher scale. Obviously not. Okay, this might be true. Like usually 5 liter reactor can give a better yield, but as you progress your KLA values, like that is the main challenge when it comes to biological systems. Your mass transfer coefficient is being affected a lot, therefore the product yield sort of tends to reduce and all. So we can't say it's more reliable than higher scale, because whatever it is, the product has to be produced in a large scale to meet the demand. So we really can't produce like this 100 meter cube by splitting it in like 5 liter or like 50 liter reduction. So option D is not nowhere related to what is being asked in the question. Option C is to produce a product of interest at large scale. Anyway, this is going to be done. So that's what a scale up talks about. For that, we'll not do any like scale down number. Mainly, like option A is to validate new operating procedures of the large production plant. Yes, you have to see some other procedures like that will be done in the your small scale reactor, that is mimicking your large scale reactor. And B is experiments are expensive to do at large scale. Exactly, that's what I was also talking about. It will be very difficult if you have like 3-4 parameters to do it in 100 meter cube reaction. It will be like waste. It's expensive, or you can call it as like waste of reagents and all. So the right answers are A and B. Moving on to question 6. What is the common unit used for rate in metabolic flux analysis? So usually this we call it as MFA, as it's related to rate. MFA. As it's related to rate, rate is nothing but something that is respective to time, change in time. So here if you see out of these 3 options, option C you can directly neglect because this is nowhere related to the time. It's not with respect to time. And the remaining 3 options are here. So we will see what is rate in metabolic flux analysis. For example, A giving rise to B, this reaction is going at the rate of V1. So we are just trying to tell that what is the change of substrate from A to B with respect to time. So mainly this is what it is. And the main thing that we can notice here is that here everywhere millimole, moles has been given. So option D also we can reject because this is not going to talk about the concentration of the cell as well as the biomass at all in the equation. In the unit that has been given. So option D is also being neglected. So here 2 options are given. One is millimole per gram and time inverse, and option B is moles time inverse. So option A is the right answer. One of the easiest way to find this is that when inside your cell no compound is present in molar concentration, everything will be in your millimole concentration value. That is one of the easiest way to find out answer A as the option. and this is with respect to the biomass also that is being produced. So moving to question 7, shear stress in a bioreactor due to aeration and agitation is because of this has been discussed in the lecture as well. It is due to the velocity gradient, as sir was giving a like example of dough, how it will be when we try to put pressure on both the sides. So that's what is like being asked here also. So if you see the options, it's not the temperature gradient, because when aeration and agitation are proper, the dead zones that we call it as, like when there is no proper mixing, then only your nutrient gradient as well as your temperature gradient might form. Whereas when there is proper mixing, these dead zones will not form at all, and this temperature gradient will not cause any shear stress in the bioreactor. So option C is not the option, and option D is presence of cells in the medium, that is not going to cause any shear stress in the reactor. And presence of oxygen in the airstream, no, it's also not going to be. As we were talking about the velocity gradient, yes, that is the right answer. Moving on to question 8. In the equation S into R is equal to dx by dt, what does R mean in the equation? So this is what we were seeing sometime before, right? Before 2 questions. R we were discussing about rate. Here R is the rate, S is the stoichiometric matrix, and R is the rate of the reaction, and dx by dt will be the accumulation term. So this is how a stoichiometric matrix looks like, and as we were telling, A giving rise to B will have a rate of V1, similarly V2, V3, V4 for B to C and whatever the reactions that are present in the metabolic pathway. So here R designates the rate vector. Moving to question 9, Mitchell's hypothesis. So we'll just see a video before talking about this.
ATP is synthesized by ATP synthase, a large complex of membrane-bound protein. Here we see ATP synthase, along with other membrane-bound proteins. Notice the large difference in the number of hydrogen ions on the 2 sides of the membrane. This difference is a hydrogen ion, or proton, concentration gradient. The energy associated with this gradient is used to synthesize ATP from ADP and PI. This occurs at the ATP synthase complex. One hydrogen ion enters the ATP synthase complex from the intermembrane space, and a second hydrogen ion leaves it on the matrix space. The upper part of the ATP synthase complex rotates when a new hydrogen ion enters. Once 3 protons have entered the matrix space, there is enough energy in the ATP synthase complex to synthesize 1 ATP. In this way, the energy in the hydrogen ion gradient is used to make ATP. Now let's watch the process again. Notice how the proton enters the ATP synthase and exits into the matrix space. Once 3 more hydrogen ions have crossed the membrane, another molecule of ATP will be made. In this example, the hydrogen ion gradient is large enough to produce 6 ATP molecules. Please watch as the remaining ATP molecules are synthesized. The process has now completed, and the result is an equal number of protons on each side of the inner membrane. Without a gradient, there is no more energy available to make ATP. In biological systems, however, a gradient is always maintained. The mitochondrial hydrogen ion gradient is generated as electrons pass through 3 membrane complexes. That process can be seen in the mitochondrial electron transport chain animation.
So Mitchell hypothesis tries to explain this osmotic hypothesis only. So based on the question that has been given here, it will be easy to visualize the ATP synthase and then see the mechanism that is happening. That's why I have given a YouTube video here. So whether did it explain related to extracellular pH and productivity? No. Intracellular pH and cell viability? No. Did it explain the ion gradient across cell membrane and the ATP generation? Yes. So when the H plus ion is passing through the ATP synthase from the outer membrane to the inner membrane, every 3 H plus ion is giving rise to 1 ATP generation. So this is what was explained in the video, and yes, Mitchell hypothesis that defines the chemoosmotic theory is related to the ion gradient across cell membrane and the ATP generation. So right answer, and then media pH and redox state of the cell, no. So the right answer is option C. Moving on to the last question, choose the correct stoichiometric matrix from below for the network shown if the material balances are done as discussed in the lectures. So here we'll just try to split it before doing the stoichiometric matrix. Okay. So here S0 is giving rise to S at the rate of R0, and S is giving rise to A with the rate of R1, and A is giving rise to B with the rate of R2. So usually your stoichiometric matrix will have rows and columns, and the rows designate the and the columns designate the the rate of the reactions. For example, R0, R1 and R2 as here, and the rows designate the metabolites that are present in the metabolic pathway that has been given to us. Here S0, S, A and B are the metabolites. So first we will see S0 with respect to R0. So in what way R0, like the rate of the reaction, is related to S0? S0 is catalyzing a reaction where S0 is the product, S0 is the reactant. So whenever it's there on the reactant side, -1 has to be given, and S0 is nowhere related to R1 and R2. That means like it's neither a product and nor a reactant. Neither a reactant nor a product for R1 and R2. Therefore, we have to enter 0 for them. Therefore, the first row should be -1, 0, 0, and coming to the metabolite S, for R0 it is a product, therefore it should be 1, and S is the reactant for the rate R1, therefore it should be -1, and it's neither a product nor a reactant for the rate R2, that is the reaction 3. So it should be -1, 1 and 0. Actually, I have written it wrong here. It should be 1. As it is in the product side, it should be 1. Coming to B, R0 is nowhere related to A, whereas for A1 it is the product, and for R2 it is the reactant. Therefore, 0, +1 and -1. And coming to B, B is not related to R0. It's not being catalyzed by R0, not catalyzed by R1 also, but S, it's a product where R2 is the rate of the reaction. So it should be 0, 0, +1. So the answer that is closest to this is option B. So these are the 10 questions that are related to week 4. I hope it is helpful in solving the assignments. Thank you so much. Tutorial 1 extended version, you guys can see that, so you guys will have a deep understanding of what each and every question corresponds to, but the sole purpose of explaining it here is just to have a quick overview of what we have seen in the last tutorial. Okay, which of the following products that are commercially produced using bioreactors? And option B, insulin, monoclonal antibodies, antibiotics, biomass. Option 3, the media used for bioprocess is contaminated with single cells with thermal response characteristics. It has been observed that by maintaining the media at 60 degrees Celsius for 25 minutes, 85% of the cells become non-viable. Determine the decimal reduction point. So the decimal reduction point is nothing but time taken for a 10-fold reduction in the viable cell concentration at a given temperature, and this is the formula. So as we have to find out Kd, we have taken this formula, T is equal to 2.303 divided by Kd, log of xp0 divided by xv, and we have found out Kd, and when we substituted Kd further in the decimal reduction time equation, we just got the answer 30.35. Okay, moving on to question 4. Write the mass balance equation for microbial cells in a bioreactor under batch operation. So under batch operation, we know that there is no inlet as well as outlet of the cells. So the overall mass balance equation will be deduced to Rg minus Rd is equal to dA divided by dt, that is rate of generation minus rate of consumption is equal to rate of accumulation of the cells. So option C is the right answer. Match the following important components of bioprocessing. Recombinant E. coli corresponds to the inoculum, glucose corresponds to the substrate, and amylase corresponds to the product. Okay. The main factors to be optimized while designing a thermal sterilization processes are based on the data that is being given here, it is very much understood that it is a function of temperature as well as time. That means like n by n naught. N is nothing but the viable cell concentration at time t, and n naught is the viable cell concentration at time 0. So this is dependent on temperature as well as time. So those two answers has to be picked for this one. And identify the type of bioreactor where aeration and agitation is brought about by pumped air alone. So when we were seeing stir tank bioreactor where aeration is brought by sparger and agitation is brought up by the impeller. Okay. Whereas in airlift bioreactor, both mixing, that is the agitation as well as aeration, is brought completely solely by pumped air alone. Whereas in packed bed bioreactor, the purpose of using aeration is only to provide oxygen and there is no mixing in this. And similarly, in the solid state bioreactor, the solid state feedstocks are being kept, and the purpose of using aeration is only to give aeration to the inoculum as well as the cells, but not for mixing. So the right answer was airlift bioreactor. Dr. A is going to do a microbial experiment in the laminar hood. Which of the following she can use to sterilize the hood? So we have seen that the answer is UV lamp as well as 70% ethanol, as UV lamp can damage the DNA of the cells as well as 70% ethanol can kill the cells by dehydration, protein denaturation. These two are being used as a combination. Match the following parts of bioreactors with their purpose. Sparger, that is used for aeration. Sparger can be used for agitation also in case of pumped air, like in case of airlift bioreactors. But in this case, in this match the followings, the sole purpose of using sparger is for aeration and impeller is for agitation, DO sensor is for oxygen level. The last question of tutorial 1 was Dr. NX runs a bioreactor experiment and found that his process produces toxic byproducts which are affecting cells viability when accumulated. Which of the following mode of bioprocess is suitable to solve this problem? Batch mode alone, continuous mode alone, fed batch mode alone, both fed batch and continuous modes. So we were saying that continuous mode is a preferred process for this because your byproducts will be continuously removed from the media, okay, and the cells will be recycled back. So you will not have cell loss, not to certain level, whereas you will be continuously like moving your removing your toxic byproducts. But fed batch is also one of the right answers because in case of batch process with a very high initial substrate concentration, what happens is that the glucose, for example, glucose being used at like 240 gram per liter or something, this will be going for what to say, all the other uh, like byproduct formation also, because already the acetyl CoA and other precursors are available in a very high concentration, so it has to be used by the cells at the appropriate time, and due to the feedback inhibition and all, it will not be going for your product of interest and it will be evenly distributed for all other byproducts also. So that's where your fed batch comes into play, that we have to use an optimal concentration of substrate concentration during the batch process, and once the fed batch starts, we will be feeding the substrate in such a concentration that it will be just utilized for the cells for the targeted product formation, but not like diverting it for others. So with this we just finished our like tutorial 1. Now we will be seeing our tutorial 2. So very good evening everyone. Today we will be seeing the tutorial 3 of BT39 NPTEL course, which is named as bioreactors. This tutorial 3 is going to help you solve the assignments of week 3, whatever the assignments are posted for week 3. So we'll just have a quick overview of what we have seen in tutorial 2, and then we will be proceeding to tutorial 3. We'll just get started. Let me share my screen. Yeah, we'll just have a overview of tutorial 2 first. So whoever has missed the last class, like tutorial 2, this will help you in getting a overview of what we have seen in tutorial 2. And in the tutorial 2 that I have explained very nicely, I have given an overview of tutorial 1 as well. So whenever I'm starting a new tutorial, I'm just giving an overview or a quick explanation of what we have done in the previous week also. Okay, so we'll just see this. Which among the following is the cheapest carbon source for microbial culture? Glucose, starch, ammonia, and glycine. So out of these, we have seen that glycine is an amino acid, ammonia is a nitrogen source, and out of glucose and starch, starch is much more like available in the nature as such. So we can use any feedstock, do any chemical treatment, thermal treatment, or else like enzymatic treatment, and then whatever the treated feedstock is there, that we can use it as a carbon source for the microbial culture. So this is a kind of cheapest carbon source that can be used for microbial culturing also. So for this, starch is the answer. So anyway, if we break down starch using any of these method, we are going to get glucose at the end. Okay, so that's why starch is considered as the cheapest carbon source. Question 2. When the substrate happens to limit growth, which among the following method is used for modeling the growth profile? So we saw the equations for all four like last time. So Ludeking Pirate model is a product formation kinetics. Monod model gives the relationship between substrate and the specific growth rate, but not any idea about the inhibition at all. Whereas Jerusalemsky-Neronova model shows that how a product can inhibit the growth of the organism, whereas Andrews and Nyok model shows the substrate inhibition on the growth. Okay, so the answer is Andrews and Nyok model. Moving to question 3, which of the following is true according to Michaelis-Menten kinetics? So Km is dependent on the enzyme concentration, Km is independent of the enzyme concentration, Vm does not vary with enzyme concentration, Vm varies with enzyme concentration. So we were seeing what is Michaelis-Menten kinetics. V is equal to Vmax, S divided by Km plus S. So here Vm corresponds to K3 into Et, whereas Km corresponds to K2 plus K3 divided by K1. So Vm varies with enzyme concentration, whereas Km is independent of enzyme concentration. B and D are the answers. So we even saw very in detail that how this derivation happens also, although sir has explained the lecture, as there is a need that we have used this equation in the previous problem. In tutorial 2 I have explained this derivation also. You guys can go to tutorial 2 and then have a look at it. Moving to question 4, uncompetitive, non-competitive and competitive inhibition. So we just saw in uncompetitive inhibition, inhibitor will bind only to enzyme substrate complex, whereas non-competitive, it can bind to free enzyme as well as enzyme substrate complex also. In competitive inhibition, it can bind only to the enzyme. Okay, so accordingly the answer is A. Among the following, which are involved in the estimation of cell concentration, as we were seeing, optical gel density, that we take it like OD 600, we take it very normally, whereas hemocytometer is used in mammalian cell cultures to find the like what to say, in a overall cell counting, mainly hemocytometer is only being used, whereas Stryfen blue, where the dead cells are like stained as blue, whereas the live cells are like intact. Okay, so Stryfen blue can differentiate between your what to say, your live cells as well as dead cells, whereas the previous one like optical gel density, it's the overall cell concentration. We can't differentiate between your live cells as well as dead cells. Okay, so the answers are like A, B, C. So moving to question 6, spirulina, it's a biomass in itself, whereas streptomycin, it's an antibiotic which comes in the secondary metabolite, whereas growth dependent products are primary metabolites. Okay. Define Ks in the monad model. Ks is the substrate concentration at which growth rate is half maximal. Ks is the substrate concentration at which growth rate is equal to 1. Ks is the substrate concentration at which growth rate doubles. Ks is the substrate concentration. Ks is the growth rate at which substrate concentration does not influence growth. So as this is talking that what happens when we put Ks is equal to S. Okay, so what happens, S and S will get cancelled, mu max divided by 2, which is nothing but Ks is the substrate concentration at which growth rate is half maximal. Moving on to question 8. In the monad model for growth kinetics, with the single limiting substrate, the plot of substrate concentration versus growth is linear, hyperbolic, sigmoidal, or cannot be determined. So as we cannot determine as such, we'll just try to compare the formula. This is the monad model equation. Anyway, it's not linear, that is for sure. Linear is what? Y is equal to mx, that is not here. Whereas in the rectangular hyperbolic, the equation is designed as like y is equal to ax plus ax divided by b plus x. It's rectangular hyperbolic. Coming back to question 9, where we have to find Km and Vmax, we have been given time S and like V. So what we have done, like we use line breaker plot. So in this, what happens, like y is equal to mx plus c. So here y axis is 1 by v and s axis is 1 by s. We have plotted the graph, and this is the equation that we got from it. 1 by Vmax is equal to this much. So we just taken Vmax, how much ever it is, and then we substituted Vmax in Km divided by Vmax is equal to 0.4577. So the answer is Km, like D. Okay. Given the above data, what happens to the value of Vm if the enzyme concentration in the system is doubled? So we already know that Vmax is equal to K3 into ET, okay, which is nothing but Vmax is directly proportional to ET. If the ET is getting doubled, obviously Vmax will also get doubled. So the answer is value of Vmax is doubled. Okay, this is what we have seen in the last tutorial. So I was just explaining you guys what we saw in the last tutorial. Now we'll get now we are going to see the quick recap of what we have seen in tutorial 3. So let me share my screen. I have already explained tutorial 4 completely, so I'm not going to go through it. Just it's going to be tutorial 3, tutorial 2 and tutorial 1. Okay. Sorry. Yes. So in tutorial 3, an industrially important enzyme is produced from thermophilic bacteria in a bioreactor operated at operated in a batch mode. 70 liter of media was inoculated using 8 gram of cells. The lab phase was found to be 30 minutes. The maximum specific growth rate of the organism is 0.52 hour inverse under the conditions maintained. Calculate the cell concentration after 9 hours of growth. Choose the answer closest to your calculated answer. Option A 10.9 gram per liter, option B 12.2 gram per liter, option C 9.5 gram per liter, and option D 0.9 gram per liter. So as it was given, x naught is equal to all cell concentration will be in gram per liter. So 8 gram of cells divided by 70 liter of media, which is equal to 0.11 gram per liter. T naught is 30 minutes, and maximum specific growth rate mu max is 0.52 R inverse, and T is equal to 9 hours. So to find the cell concentration after 9 hours of growth, we know the formula 1 by mu ln of x divided by x naught plus t naught is equal to t. Substituting all the values, if we proceed, we just get the value of x is equal to 9.2 gram per liter. So question 2, in a chemostat containing suspended cells operating at steady state condition with negligible death rate, which of the following statements is or are valid? So specific growth rate of cell is equal to the dilution rate. Specific growth rate of the cells is greater than the dilution rate, specific growth rate of cells is lesser than the dilution rate. Specific growth rate is unaffected by the dilution rate. So we know that in a chemostat, like we'll have what to say, your media flowing into the chemostat, whereas at the same time the fermentation broth along with the cells will be removed from the fermentation broth also. If you see the cells, there won't be any inlet of cells, it will just get removed. Therefore Ra is equal to 0, and as it was mentioned in the question that negligible death rate, the consumption is also equal to 0. Therefore minus R0 plus Rg is equal to 0. Like Rg is equal to R0, and Rg we know the generation corresponds to the specific growth rate into cell concentration into volume, which is equivalent to rate of outlet of cells is equal to F into X. So if you just rearrange, you get X is equal to 0, mu is equal to D. Therefore, the specific growth rate of cell is equal to the dilution rate. If E. coli is grown on a media containing both glucose and lactose, which of the following phenomena is observed in its growth? So dioxy growth or immortality, whatever is given. So we know that this is dioxy growth because at first the organism will try to take glucose, and then once the glucose is completely exhausted from the media, then it will start consuming lactose. That's why there will be a second lag phase, because it's getting adjusted to the environment, that is the lactose being present, and then the exponential phase starts. So in addition to this, if glucose and lactose both are present, I wanted to explain the molecular mechanism. So the CAMP levels will be very less. Therefore, the CAMP cap complex will also be very less. That corresponds to what you know, like it will not go. This is an activator for this lac operon. It will not go and bind. Therefore, your transcription will not happen. Whereas when only the lactose is present, when the glucose is completely exhausted, like it's vice versa of what we talked before. CAMP level will be very high, therefore CAMP cap complex will be present in higher concentration. It will go and bind to the upstream regions of this lac operon, therefore the transcription happens. At which phase of cell growth is maximum specific growth rate observed? So we know that during like lag phase, mu is equal to 0, and stationary phase, mu is equal to 0, and the maximum specific growth rate happens during the exponential phase. Moving to question 5, what happens if the dilution rate is larger than the D critical, that is critical dilution rate? So we already know that critical dilution rate is equal to feed flow rate divided by volume. So if we cross the, if D is greater than D critical, what happens is that the flow rate is very, very high compared to whatever the critical that has to be maintained. Therefore, the washout of cells happen. Moving to question 6, Pseudomonas species is cultured in a chemostat for the production of lipases. The chemostat has a capacity of 45 meter cube, volume is equal to 45 meter cube. Substrate concentration in the SA is equal to 14 gram per liter, and Ks is equal to 0.51 gram per liter, and maximum specific growth rate is 0.36 R inverse. So we have to estimate the feed flow rate required to achieve 90% substrate conversion. So it has already been written in the given, and to find F, mu is equal to mu mass S divided by Ks plus S. As we already know all the formula, we are just substituting and finding out D. In the previous question also, we just saw in chemostat mu is equal to D. Therefore, D is calculated, and D is equal to F by V. As we know the capacity, that is 45 meter cube, we can just back calculate, which is 18.88 meter cube R inverse. Moving on to question 7, sulfide oxidation method is roughly used to estimate the volumetric mass transfer coefficient, because how much, based on the RPM and the flow rate that we are setting in the fermentation in the bioreactor, the oxygen will completely will be used as a dissolved oxygen, and then if dissolved oxygen and sulfate reacts, that will be converted into sulfate. So with this conversion, we can back calculate what is the volumetric mass transfer coefficient. Identify the device used in the air flow rate measurement in the bioreactor. It's not baffle, it's not RTD, that is related to temperature. Tachometer is related to your power that is used for the motor rotation. So the answer is B, rotameters. Moving to question 9, we have to match the following. Thermophile is nothing but that grows at a very high temperature, which is 45 degree to 80 degree Celsius. Psychrophiles are something that is at lower temperature, 0 degree to 20 degree Celsius. Mesophiles at normal temperature, that is 20 to 40 degree Celsius. If the substrate concentration, that is S, is very greater than the Ks, then which of the following is true according to the Monod model? A, the organism grows at its maximum specific growth rate. The organism specific growth rate of the organism decreases. The specific growth rate of the organism doubles. The organism reaches the stationary phase. So if S is very greater, which means Ks is very negligible, so we can neglect it. S and S will get cancelled. Therefore, mu is equal to mu max. So the right answer is the organism grows at its maximum specific growth rate. So we have discussed tutorial 4 in detail in this lecture, and then we have almost had a recap of tutorial 1, tutorial 2 and tutorial 3. So this whole video is going to help you in solving the having a recap of all the 4 tutorials that we have seen till this point. So if there are any questions further, you guys can send a email, or else like you can post it in the chat box or something. I'll try to get back to you guys. Thank you.